Hello everybody, so today we're going to be going over the fungal lab that you guys would have been doing normally if you were at campus. However, I'm going to be doing it for you so you guys can follow along and see what you would normally be doing if you were able to do this at campus. So we're going to be working with three different types of bacteria today. The first one we're going to be working with is called Pilobolus crystallinus, and this is known as the shotgun fungus. This is the fungus that, thanks to osmotic pressure in the spores, literally shoots out spores once they've reached maturity and they're ready to expel themselves. So in this lab, what we're going to be doing is we have several growth plates that have a rabbit dung agar on them, and we're going to be transferring 1 centimeter cube sections of the bacterium from the culture plate onto the new plates. We're going to be inverting cups over the top of them like this, putting aluminum foil on the sides, and then placing tape over a hole cut on the top. And this hole is going to correspond with a hole in the bottom of the cup. So what it's going to look like is we're going to have the open growth plate with the transferred bacterium, which isn't on this one just yet. And then after about a week of growing in a room temperature room, they will eventually reach the point where the osmotic pressure will get high enough that they'll shoot themselves up, and we'll be able to see it over that tape that we've taped there. So now I'm going to walk you through the steps on how you would actually be doing this if you were in lab. All right, so first things first, I have everything labeled ahead of time. As you can see, I have 3 replicates, and on the sides of the replicates, I have labeled the date as well as what number they are. This is going to be replicate 1, and that's the one where I'm actually going to show you guys how we're doing this. On the left here, you see that we have the culture plate that originally came with the bacteria, Pilobolus crystallinus. And now what we're going to be doing is we're going to be taking a 1 centimeter cube from here and transplanting it over here and allowing that to grow for a few days. So the way we do that is we remove this cover, exposing the bacteria. We take our scalpel, and now we're going to cut a 1 centimeter cube away from this culture plate. So we'll get this nice group right here. So now that we have the culture on the end of the scalpel, we're going to transplant it to this other plate, just like so. So some things to keep in mind that are general hygiene standards. Don't touch the cutting edge of the scalpel to the actual growth plate. That's an avenue of way we can contaminate the culture plate. We're going to dispose of this in an autoclave bag after this, and we're going to safely cap it, which I'll show you right now. So, as you can now see, the scalpel is safely capped in its disposable sheath, and we're going to dispose of this in an autoclave bag after we're done with the experiment. But now what we're going to want to do is place a lid over our culture like this, push gently down to make sure that it locks in place. After we put the lid on top, what we're going to do is get this aluminum foil, place it over the top like this, and we're going to use our pencil to poke a hole in the top like I already did. This is the hole that the spores are going to erupt out of. And now what we're going to do is, much like these over here, which you can see, have tape over these holes. So now we're going to replicate that over here. All right, so we've placed the tape over the hole. The reason we're using tape, and the reason it is clear tape in specific, is because this way, when the spores erupt out of that hole, we'll actually be able to see them caught on the tape itself. Now we're going to place these under strong direct light for about a week. According to your lab manual, if we leave it under strong light for about 2 to 3 days, then at 11 to 12 a.m. on the day of them being first evident, they'll actually explode. And if conditions were quiet in the classroom, you'd actually be able to hear them spurting out and hitting the tape. Alright guys, so on to the next experiment. Now this one isn't in your lab notebook. However, this is an interesting experiment with something that people don't really associate with fungi, but bioluminescence, something that very much so is associated with fungi. So today we're going to be looking at a culture of Armillaria mellia. So this stuff looks really wild, as you can see. And what we're going to be doing is very similarly with the other bacteria that we're using on the rabbit dung agar plates. We're going to be taking a 1 centimeter cube using a sterile scalpel and transplanting that from the actual culture to new agar plates. And this one's going to take a little bit more time to grow. So according to the instructions for this one, new hyphal growth can be observed in about a week. Rhizomorphs can take about 10 to 14 days extra after that. However, once you see those rhizomorphs, that is when we can start seeing a bioluminescence. So maximum bioluminescence occurs at about 3 weeks, and it can last for as long as 10 weeks under correct environmental conditions. So this one is one that we're going to start here in lab, and much like what we're going to do with the rabbit dung bacteria, we're going to show you pictures of it once it's finished in about a week or so for the rabbit dung. In this case, in about 2 weeks, you might start seeing pictures. It depends on how well these do. And as with the rabbit dung culture plate, we're going to be doing 3 replicates. I already have 2 done over here. We're going to be working with the 3rd over here. And as with the rabbit dung, I've labeled them with the date and the replicate number. And you can see that I've already implanted some of the spores right there. It's harder to see on this one because of the moisture. However, now we're going to get into it. So, as with the rabbit dung, we're going to be taking a 1 centimeter cube from this initial culture plate, and you can even start see the areas where I took the other 1 centimeter cubes from. So let's go ahead and get started. Now that I have the 1 centimeter cube of fungus, I'm now going to transplant it to the waiting growth plate, just like so. So remember, we're going to invert it so that the actual fungus is touching the new growth plate. General aseptic technique: do not touch the new growth plate, and do not touch the new growth plate with the cutting edge of this scalpel. That is avenues for contamination and could interfere with this entire process. And much like with the other scalpel, this scalpel is going to be capped and disposed of in an autoclave bag as well. So, scalpel safely capped. I'm going to set this over here to the side with its packaging, and I'm going to dispose of it later. So now there's only one experiment left to do, and that is going to be viewing the penicillin under a microscope. And for that, we're going to need to go look at a microscope that is hooked up to a computer. And a few more notes to make on the growing conditions. Just to reiterate, in case there was any confusion, the rabbit dung agar plates seen here are going to be grown under intense light at room temperature for about a week. And that is what will get the best growth out of those. However, for the bioluminescent bacteria, the Melia, that is going to be grown in basically a darker room as we can get, but it's also going to be at room temperature just like the other one. However, unlike the other one, this one is going to be grown in the dark.